Hi again, and welcome to part 3 of this video series. In this video, we'll show you how to define regions of interest and how to extract nuclei labels from your input images. In order to do so, we will be covering steps 1 and 2 of the pipeline workflow. Remember, there are a couple of ways to start your workflow. In alternative 1, you begin by manually selecting your regions of interest, drawing directly on the input image. Then the Stardist model is run only within those ROIs, which makes the process more efficient by reducing the area the model needs to analyze. This is especially useful when working with large images or when only specific regions are relevant. Regardless of the approach you choose, all ROIs are saved to disk in your input raw data folder. This not only supports reproducibility, but also lets you reuse or revisit those ROIs later in your analysis. In addition, keep in mind that if you want to analyze your entire image, you can skip the region selection step. As you know from the previous video, nuclei label prediction is a time-consuming task, but it can run unattended. In alternative 2, you start by predicting nuclei labels for the entire image. This approach is especially helpful if you haven't had the time to draw your ROIs in advance. After prediction, you can then draw the ROIs at your convenience. Once the ROIs are defined, you can apply them to the full image label map using the notebook 002 ROI on full image nuclei labels. This allows you to extract or analyze only the relevant regions from the already predicted full image labels. Let's start by showing you how to define your regions of interest. First step is opening VS Code and navigating to the repository. You just use File, Open Folder, and then you select the location where you clone the repository. In my case, I clone all of my repositories under GitHub repos. Then you choose brain tissue nuclear segmentation and select this folder. Afterwards, this is how you would see the folder structure. You just click on Stardist and open the 001 batch processing draw ROIs notebook. This is the first time that you open this notebook. You would have a prompt here saying select kernel. This is where you choose your virtual environment of choice. In my case, I'm using Brain Nuke Stardist, which is the environment that you can create following the instructions in the repository. And the first step is importing the tools and utilities that this notebook needs. This takes a few seconds the first time that you do it. And the next step is defining what are your input images. In my case, I have some test data that is sitting one level above this notebook. So I'm using relative paths, but most probably you would need to use absolute paths. So you would look first to where your data is located. In my case, I have some images again under test data. So just double click on it, and once you're inside the folder containing your images, you copy this path. Once you have copied it, you paste it in between the quotation marks and place an arrow in there. So now you should see the images that are contained within that folder, and in the next cell we are going to open them. So we want to open the first image. You should put a 0 in there. So 0 defines the first image in the directory. This is because in Python 1 starts counting from 0. If you wanted to open this second image, you would put a 1 in there. The next variable that we need to define is the slicing factor. So in order to explain this, we will go back to the presentation. The slicing factor helps reduce memory usage by downsampling your images. Often, images are captured at much higher resolution than what's actually needed for analysis. By lowering the resolution, we reduce the computational load and speed up processing. If we set up the slicing factor in the XY plane to none, the full resolution image would be analyzed. A slicing factor of 2 means we analyze every second pixels, 4 means every fourth pixel, and so on. But be careful, too much downsampling can cause important details to be lost. In the SF4 panel, for example, sparse nuclei still appear well defined and are easy to segment. However, in denser areas, this loss of resolution can make it harder to accurately detect and separate nuclei. The same principle applies to the set dimension. Often, we acquire more optical slices than necessary for effective analysis. While this can improve visual detail, it also places a significant load on system memory and processing speed. By applying a slicing factor along set, we can reduce the number of slices processed. just like in the XY plane. No matter which slicing factors you choose, whether in X, Y, Z, or both, it's essential to keep them consistent throughout your entire pipeline. This consistency ensures that all processing steps operate on data with the same resolution and scale. This is especially important when training a Stardist model, since mismatched resolutions between training and inference can lead to poor performance or inaccurate predictions. Okay, so now that you're familiar with the slicing factor concepts, we are just going to load our first image with no slicing factors, so we are loading the full resolution image. And since I have a couple of windows, I'm just going to open it to the side. So now we can start exploring our images in Apari and navigate across the different channels. I would use this channel to define my regions of interest. In order to do so, you create a new labels layer. You name it according to the nomenclature that you're using. In my case, I will define the dentate gyrus. And then you select the brush tool. I zoom in using the scrolling wheel from the mouse and define my region of interest by holding the left click. Once I'm done, make sure you're connecting, because the next tool that we are going to use is the fill bucket. So that would be one region of interest. You see that if we scroll across channels, you cannot see it. So in order to solve that, the only thing that you need to do is create a new labels layer. And this time you increase the number of edited dimensions to 3. So now your label, the one that you're painting, will propagate across all channels. Use the fill bucket, and then you see that if you pan across channels, your label is still there. Then I will define a second region of interest by creating a new labels layer. I'll call it CA. And I repeat the same process. I choose the brush. I'm just changing the label number, so it's easy for me to keep track of different regions. And then define this new region of interest. I make sure these are closed. Use the fill bucket. And now I have defined a couple of regions of interest. You see that the number of edited dimensions for this one is 2. So if I move across channels, that one will disappear while the other will remain. Okay, so now that I have defined a couple of regions of interest, the next step is saving them. And in order to do so, you just scroll down and hit play on the last cell. It will close the Apari window, and then it will say the following labels would be analyzed. In my case, I painted 2 regions of interest, DG and CA, and now this would be stored under the input folder, which in this case it was test data. You'll see a new folder there called ROIs, and then one folder for each of the ROIs that you have painted. It is very important that you keep using these very same names as you paint across your images. So now I will open the second image by putting a 1 in there. I'll load it in Apari, and now I will paint the same regions of interest, but on a different image. So I'll paint DG first. It's very important that I'm using caps. Choose the brush tool, fill it, and then for the second region, I name CA. I change the label to keep track of it. Brush tool again. Fill. Now we move back to the Jupyter notebook. Hit play, and then we are saving these regions again. Now you would see that for each image we have defined CA and DG, and they are stored here. If you want to see them under the Windows Explorer, you just right click there and say reveal in File Explorer, and you will find your files in there. So these are the regions of interest that you have painted already. At this point, you can keep exploring your dataset and painting your region of interest over all images by repeating the same process. You change the index there, you hit play, annotate the images, hit play, and then you save them. Once you have finished drawing all your regions of interest, you can continue with the next notebook that is in charge of predicting nuclei within these regions of interest. Alright, so let's continue with the workflow. Now that we have defined all the regions of interest, we can move on to the nuclei prediction step within those ROIs. Remember that if you haven't defined any ROIs, you can skip straight to this notebook. It will generate nuclei labels for the entire image. Before starting to batch predict nuclei labels, it is important to test how the Stardist model performs. We can make use of the single processing mode of predict nuclei labels to do so. Now let's switch back to VS Code. Okay, so now we have generated our regions of interest and they are saved under our raw data folder, in this case test data. So now we want to evaluate how the different Stardist models perform at generating our nuclei labels of interest. In order to do so, we'll use the single processing mode of predict nuclei labels. The first step, as in the previous notebook, will be to choose the environment that we need to perform the tasks, load the tools and utilities that we need for this notebook. This again takes a few seconds the first time that you do it, and then define the data directory that contains our images of interest. In this case, it's already defined. We see this couple of images that we use to draw our ROIs. Then we will open the first image in our folder to check the channel position of the nuclei labels. I'll just move it to the side. And here we can explore the different channels in the image. And we can see that the nuclei labels are in the last channel. So this would be channel 0, 1, 2, and 3. So the nuclei are present in the channel number 3. We have already covered the concept of slicing factor before. So the next variables that we are going to define are the nuclei channel position, in this case is 3, that's already defined. Then the number of tiles, so we'll go back to the presentation to define the concept of tiling and why it's important. The concept of tiling is tied to how Stardist generates the nuclei labels. Stardist prediction has two main phases. Phase 1 involves a neural network prediction. The input image is normalized and passed through a U-Net to generate polygon candidates for each nucleus. This step can be GPU accelerated via TensorFlow and benefits from tiling when working with large 2D or 3D images that doesn't fit in GPU memory. The nTiles parameter defines how the image is split into overlapping tiles in Z, Y, and X axis to allow processing within memory constraints. We will show an example in the next slide. Tiling is useful for large images that don't fit in GPU memory and cause out-of-memory errors. Ideally, use nTiles of 111 to process the entire image at once. However, this often requires trial and error. Start with nTiles of 122 or 144 and increase as needed until the image fits into the GPU VRAM. For example, let's look at the case of nTiles of 122. This means the image will be divided into one tile along the Z axis and 4 tiles along both the Y and X axis. Phase 2 involves post-processing via non-maximum suppression or NMS. Once predictions are made, Stardist runs an NMS step to filter overlapping detections using their associated object probabilities. This step is not GPU accelerated and runs on the CPU over the entire image, scaling with the number of cores. For large images with many nuclei, this step can become a bottleneck. Okay, next we have to choose the segmentation type that we want to perform on the input image. Choose 2D if your input is a single plane, multi-channel 2D image. The output will be 2D nuclei labels matching the input format. Choose 3D if your input is a multi-channel set stack and you need to extract 3D information by generating 3D nuclei labels. If your 3D stack contains a single cell layer and 3D context isn't needed, you can process it as a maximum intensity projection. This significantly reduces computation time and resource usage. The output will be 2D nuclei labels. And then we need to define our Stardist model to perform the segmentation task of the nuclei. If we are using a segmentation type of 2D, we will need to have an available 2D Stardist model, or if we are performing a 3D segmentation, we'll need a 3D Stardist model. If you don't have access to pre-trained models, just type test and then a standard Stardist model will be loaded. Now let's explore a few of the situations that you might encounter and also the different segmentation types that you might need to use. Just as a side note, you will see this cell expanded normally, but since you don't need to input any data in there, you can just collapse it by double clicking on it. Okay, so let's say we are going to process a 3D stack and we want to obtain 3D labels. So then we have chosen segmentation type of 3D. As we mentioned before, we want to fit it in the smallest style possible. So we'll try with a style of 111. And try and run the Nuclei prediction. As we can see, we have obtained an out-of-memory error. So this means we need to chop our image into a larger number of tiles. Let's try with a tile of 1, 4, 4, just to be on the safe side. So while this is predicting, and if we open the task manager, we should see a peak in GPU usage corresponding to the generation of candidate polygons for each nuclei, and afterwards GPU usage will drop and we'll see 100% utilization of the CPU during the non-maximum suppression step. If we take a look at the resource monitor, we should see all the nuclei of the CPU working at 100%. This means the environment is correctly configured, and then the non-maximum suppression step is making use of multi-threading. If that is not the case, I'll share a link in the video description to an image.sc post describing how to solve these issues. Okay, so now that the Nuclei prediction step has completed, we see there's no usage of the GPU. The VRAM is still occupied by the Stardist model, and then there's a decrease in the usage of the CPU. Then we can see that the Nuclei prediction has also been completed. And then we can explore the Nuclei labels generated across the different slices. If we click here, we toggle to the 3D view. And we can see how this model has correctly identified this multi-layer structure and also its nuclei boundary. If we were to use a different model, like for example the standard Stardist 3D model, by typing test here, we would observe a very different set of results. So this model is not tailored to this input data, and then we'll obtain subpar predictions. The nuclei predictions that we have generated before will be stored under your input folder containing your images, under nuclei predictions, your segmentation type, 3D in this case, and the model name, MEC01, and then also on an ROI basis. After running the standard Stardist 3D model, we can observe how the prediction is subpar. It's missing a lot of the nuclei, and some of them are not even correctly segmented. Let's imagine that now we want to process our 3D stack as a 2D maximum intensity projection. So we'll just type 2D here. We'll have to input a test model name because there's no pre-trained 2D model. Apply these changes and then run the prediction again. As you see, running the predictions in 2D takes much less time than compared to 3D. But again, since we are using a standard Stardist model, it's generating subpar results. It's missing some of the nuclei, and it's also incorrectly detecting nuclei substructures. Since we have chosen 2D segmentation type, the output are 2D labels. Finally, and after running all of this notebook, you might see that your RAM memory is getting full and you might be getting resource exhausted errors. In order to solve that, the best is to restart the kernel. And then you'll see how the VRAM GPU is liberated. Same thing for the RAM. And it doesn't hurt to close VS Code to liberate some extra RAM and open it again, just to start freshly. Okay, so now we have defined a model that works for our dataset and some tiling strategies. Let's predict nuclei in batch prediction mode. We'll just open the notebook 002 batch predict nuclei labels. We will load the tools that are necessary to run this notebook. Then we will define the directory path containing our data. And since we have already run this in single processing mode, we know that we are using no slicing, that our nuclei channel is in position number 3 according to Python, that we should use end tiles of 144, and then we want to run a 3D segmentation with this particular model name. So we just hit play. And we'll start predicting the nuclei. Since we have already run one of the images through the single prediction mode, the notebook notifies you that nuclei predictions have already been found for that particular image, so it's not going to generate them. For the second image, it's going to generate them, as you can see here. Okay, so at this point, the nuclei predictions have been completed for the two images present in our test dataset, and we can see them under 3D, MEX01 model name, and then its different ROI. We can see them there. If we want to explore the nuclei labels that has been generated during the batch prediction mode, we can just go back to the single processing mode and explore them individually. In this case, now I'm going to change the index to 1 to open the second image in the folder. I'll just move it to the side so we can visualize it. This is the second image, and then I make sure that all the variables are correct. Nuclei channel position, number of tiles, segmentation type, model name. Everything seems fine. So when I hit play, this notebook is going to inform me that the nuclei predictions have been found, so it doesn't need to generate them since they have been created during the batch processing mode. And then you can explore how they look. Okay, so everything seems to be working just fine. At this point, we are done with batch nuclei prediction. Finally, let's cover the alternative batch processing workflow. In this workflow, you first predict nuclei labels for the full image, and then you apply your predefined ROIs afterwards. Let's switch back to VS Code to see how this looks in action. Okay, so the first thing that I need to do is trick the pipeline into thinking that there's no ROIs defined. So I'm just going to change the name and include an underscore in there. Now I'll open batch predict nuclei labels. I'm going to load the utilities, point to the directory path. I'm defining a slicing factor of none. Nuclei channel is 3, 3D segmentation type, model name, the same as we have tried before. And now we'll start the batch prediction for the full image. And now, since the pipeline is not recognizing any previously drawn ROI, it's going to predict nuclei labels for the full image. Okay, so now that the full image nuclei prediction for both images has been completed, I'm going to revert the ROI folder to its original status. I'll just remove the underscore, and I'm going to open the batch processing ROI on full image nuclei labels. This will mask the full image nuclei labels with the ROIs that are present on the disk. I'll just have to select the kernel, load the necessary tools. I have pointed to the directory path that contains the data, and I am going to define the same segmentation type and the same Stardist model. Now it will say that the following region of interest will be applied over the full image, CA and DG. And at the end of this process, all nuclei labels for each region of interest will be generated and saved to disk. So now under nuclei predictions, in addition to the full image, we can see that the CA and DG regions have been generated.